Marine biology as a discipline on every coast of the world. Scientists work in field locations and in marine stations ranging from multi-million-dollar structures to small shacks with fanciful paintings of lobsters and crabs above the door. Some put out to sea in large ships, whereas others scarcely wet their knees. The purpose of this textbook is to give you an organized way of turning a fascination for the sea into an appreciation of the principles of marine biology that reflect the function and ecology of marine life. Snorkel on a coral reef and you will see coexisting schools of large numbers of species of fishes. But why do so many species coexist in a very limited space? How do all these creatures interact to form the seascape? Such questions require an organized approach to a complex and somewhat foreign world. By the time you have finished your course and this textbook, you will be more familiar with that world. You will also be familiar with many human activities that put this world in peril, which makes an understanding of marine biology extremely important. Marine biology is a subject mixing functional biology and ecology. Marine biology is a diverse subject, but its main elements are functional biology and ecology. Functional biology is the study of how an organism carries out basic functions such as reproduction, locomotion, feeding, and the cellular and biochemical processes related to digestion, respiration, and other aspects of metabolism. Problems relating to function are quite varied. They might deal with questions such as, when a whale dives for food to very great depths, how does it conserve oxygen? Ecology, on the other hand, is the study of the interaction of organisms with their physical and biological environments and how these interactions determine the distribution and abundance of the organisms. For example, why do so many species requiring limited space coexist on a coral reef? Why doesn't one superior species win out and displace the others? Because ecology is an environmental subject, the field of marine biology must cover the basic aspects of marine habitats. We shall therefore spend considerable space explaining the various seascapes that are important to marine life. Biodiversity is the third major factor in marine biological studies. Marine environments can be very rich in species but vary tremendously in the number of species, or biodiversity. Coral reefs may contain thousands of species, but a rocky shore in high latitudes may contain fewer than 50. How do species arise? Why is there variation in species numbers from habitat to habitat and from time to time within a given locality? What is the consequence of living in a very diverse community? How can so many different species make a living in a coral reef? Functional biology, ecology, and biodiversity, as you will see, are very interactive components. Historical background of marine biology. Marine biology began with simple observations of the distribution and variety of marine life. A native lore of the biology of the sea has accumulated over thousands of years by those living near the shore and by fishing peoples. The earliest formal studies in marine biology date back to a time when there was little distinction among scientific specialties. Early biologists were natural philosophers who made general observations about anatomy and life habits. We owe the beginning of this tradition of natural philosophy to Aristotle and his Greek contemporaries, who recorded their observations on the distribution and habits of shore life. Aristotle described the anatomy of the octopus and other marine creatures, noticed that some sharks give birth to live young, and observed that some whales have structures that resemble hog bristles instead of teeth. The next major steps forward took place in the 18th century, when a number of Europeans began to observe and classify living creatures. Most prominent among these was Linnaeus, who developed the modern means of naming species. He described hundreds of marine animal and plant species and developed larger-scale classifications. The great French biologist Georges Cuvier classified all animals into 4 major classes of body plans: articulata, radiata, vertebrata, and mollusca. The 18th century was an important era of oceanic exploration. A number of expeditions circumnavigated the globe, bringing glory to explorers and new Pacific territory to European nations. Many of these expeditions had scientific components as well, and scientific staff was charged with collecting terrestrial and marine plants and animals. The voyage of French Captain Nicolas Thomas Baudin explored the tropical Pacific, and numerous marine mollusks were returned to France. Captain James Cook supervised the mapping of eastern Australia in 1770, and his scientific staff collected biological specimens all over the Pacific Ocean, which became the foundation of large collections in Great Britain. Until the 19th century, most marine biology consisted of the description of anatomy and the naming and classification of species. Little was known about function and ecology. The only knowledge of open ocean life was confined to experience with animals that were fished or observed in the open sea. By the early 1800s, however, the study of natural philosophy had become popular, and a number of brilliant individuals devoted their lives to studying the ocean and its denizens. In the 19th century, marine biology developed into a science involving ecology and hypothesis testing. Edward Forbes of the Isle of Man was the first of the great English-speaking marine biologists. After failing at art and abandoning his medical school studies, he set out to sea and participated in a number of expeditions in which a bottom dredge was used to dig into the seabed and collect organisms. He was the naturalist on the Beacon, a ship that sailed on the Mediterranean Sea. Forbes found that the number of creatures decreased with increasing depth and then proposed what was probably the first marine biological hypothesis, or testable statement about the world of the sea, the azoic theory, which stated that no life existed on seafloors deeper than 300 fathoms. Forbes also discovered that different species live at different depths, and he proposed that the broader the depth zone of a species, the wider its geographic extent. Forbes opened up the ocean to scientific research and was appointed to the most prestigious post in natural philosophy of those times at the University of Edinburgh, Scotland. He published maps of geographic distributions of organisms along with a natural history of European seas. He inspired countless followers to an interest in natural science. During this time, many great pioneers from a number of European countries joined Forbes. In 1850, Norwegian marine biologist Michael Sars disproved the azoic theory by collecting and describing 19 species that live deeper than 300 fathoms in Norwegian fjords. His work inspired a new interest in deep-sea biology. The first plankton net was used during this period, and crude submersibles were developed. Marine biology was on its way. Although he is usually remembered for his theory of evolution by means of natural selection, Charles Darwin is the other great English father of marine biology. As a young man, he worked as naturalist on the HMS Beagle, which sailed around the world in the years 1831-1836. He later wrote The Voyage of the Beagle, one of the best-selling travel books of the 19th century. Darwin made extensive collections of marine animals and concentrated his own later efforts on the classification of barnacles. While on the Beagle, Darwin formed a theory of the development of coral reefs. He pictured the growth of coral reefs as a balance between the growth of corals upward and the sinking of the seafloor. If Forbes's azoic theory was the first important marine biology hypothesis, then Darwin's coral reef theory was the second. This subsidence theory was published in Darwin's first scientific book, and its brilliance was immediately recognized. Previously, most had believed that coral reefs in the open Pacific developed from the colonization and growth of corals on submerged extinct oceanic volcanoes. In contrast, Darwin argued that coral reefs developed around emergent rock that was slowly sinking, and this downward motion was balanced by upward growth of the corals. In this subsidence theory, as applied to the development of atolls, Darwin was proven correct. About 100 years after the theory was developed, scientists drilled a hole in Eniwetok Atoll in the Marshall Islands of the Pacific and bored through hundreds of meters of coral rock before hitting the volcanic rock basement below. Since reef corals can grow only in very shallow water, this finding proved that the reef had been growing upward for millions of years as the island was sinking. Darwin was not completely right about coral reefs, however, insofar as he theorized that all reefs in the world are stages of subsidence leading to atolls. This has proven to be wrong. Many reefs are not subsiding, and atolls are special cases of reefs on volcanoes rising from oceanic crust. Fisheries research began in earnest in the 19th century and became central in marine biological research. Such research was also the beginning of applied marine biology and was necessitated by a need to understand how to find and manage populations of fish. England was first at this activity in 1863. Many nations began research efforts later in the century. In the United States, the Fish Commission sought to relate characteristics of the oceanic environment to the life history of fishes. Marine ecology became synonymous with fisheries research, and Canada used a fisheries emphasis to develop distinguished laboratories on both the Atlantic and Pacific coasts. W. B. Carpenter and C. Wyville Thomson led a major expedition in 1868-1869 that foreshadowed the later Great Challenger expedition. Both had a passion for marine biology, and they convinced the British government to outfit the Lightning, a steam and sail-powered ship that dredged the seabed of the northern waters of the British Isles. Like Norwegian biologist Michael Sars, they found marine life deeper than 300 fathoms and thus also helped disprove Edward Forbes's azoic theory. The deep maintained its allure to marine biologists, as it was thought to be a museum of living fossils because many animals such as stalked crinoids were found in ancient fossil deposits and in deep water in the living ocean as well. They also found distinct bodies of water with different temperatures, which was an early discovery of the distinctness of some oceanic water masses. The voyage around the world of the HMS Challenger gave us the first global-scale view of marine biology. These expeditions set the stage for the Great Challenger expedition that circumnavigated the globe and provided the first global perspective on the ocean's biotic diversity. The voyage was led by C. Wyville Thomson and by the great naturalist John Murray. The Challenger sampled the waters and bottoms of all seas except the Arctic. After the expedition, 50 volumes were needed to describe the tremendous number of organisms that were recovered. On this expedition, chemist John Buchanan was able to disprove the existence of a so-called primordial slime on the seafloor, called Bathybius, which was supposed to be capable of giving rise to higher forms of life. The famous zoologist Thomas H. Huxley had published a well-known paper claiming that a whitish material found in samples from the seabed was evidence of the presence of a material that was continuously giving rise to life forms. This idea was very controversial. Buchanan discovered that the slime, which had been observed in collected samples of seawater, was merely an artifact of preserving seawater with alcohol. The chemical reaction of seawater with alcohol resulted in a white precipitate, which Buchanan claimed was previously mistaken by Huxley to be the mysterious Bathybius. He therefore falsified a major claim about the continuing origin of life on the seafloor. This discovery fit well with Louis Pasteur's earlier conclusion from lab experiments that life did not just spring from inanimate substances. The explanation for the white slime named Bathybius still remains an open question. Toward the end of the 19th century, marine stations began to spring up over the world, starting in 1875 with the Stazione Zoologica in Naples, Italy. This station set a pattern of international participation by the scientific community. In the 1880s, marine stations were established in England and Scotland. During the same years, Prince Albert I of Monaco outfitted several yachts and larger ships that sampled the ocean, and in 1906, he eventually founded an oceanography institute and museum in Monaco. This facility came to be directed by famous inventor oceanographer Jacques-Yves Cousteau, who died in 1998. In America, zoologist Alexander Agassiz led oceanographic expeditions, was the first to use piano wire instead of rope to lower samplers, and studied the embryology of starfish and their relatives. The now famous Marine Biological Laboratory was founded on Cape Cod in 1886, and a number of marine stations were founded in Europe toward the end of the century. By the turn of the 20th century, marine stations existed in many European countries. Marine laboratories such as the Friday Harbor Laboratories in Washington state made their appearance in the United States soon thereafter. Marine biology was now a full-fledged science with a proud history of exploration and theorization. Advances in modern marine biology included the development of major research institutions, faster ships, better navigation, and greatly improved diving technology. The early part of the 20th century witnessed the founding of great ocean-going institutes and a new technological ability to explore the ocean to its greatest depths. In America, the founding of the Scripps Institution of Oceanography in Southern California and the Woods Hole Oceanographic Institution on Cape Cod gave the United States a unique ability to study the open sea. A large number of open sea expeditions expanded our knowledge of marine life. The voyage of the Danish Galathea was the last great deep-sea expedition of this era. As had happened in Europe toward the end of the 19th century, marine stations were opened in every coastal state in America. Marine biology also flourished in many universities. Our knowledge of the ocean expanded during World War II owing to the need for more navigational information. Advances in navigation, deep-sea bottom drilling, remote sensing, and other techniques led to a great expansion of our knowledge of the sea. A rich diversity of open ocean and shore biological research has since flourished to the point that scores of journals now record the activities of a community of thousands of scientists. The number of such scientists in 1850 could have fit comfortably within a single room. Technology in both the laboratory and the open sea has played an important role in the development of marine biology. Before the 19th century, poor navigation, inadequate sailing vessels, and generally crude bottom dredges and plankton nets prevented researchers from sampling the ocean systematically or completely. By the late 1800s, however, steam vessels allowed for the rapid lowering and raising of samplers, and navigation was better. In the 20th century, modern diesel-driven ships such as the RV Nor, ported in Woods Hole, Massachusetts, could navigate accurately by means of satellite navigation. Before the mid-20th century, the deep-sea bottom could not be seen unless a piece of it was dredged and brought to the surface. This has changed dramatically owing to the development of manned submarines, remotely operated vehicles, and scuba diving. William Beebe pioneered deep diving when he descended in a metal sphere, the bathysphere, to a record depth of 923 m in 1934 off the Bermuda coast. In 1960, the spherical steel bathyscaphe Trieste made a spectacular descent into the deepest oceanic trench off the Marianas Islands in the western Pacific Ocean. By the 1970s, a number of submarines routinely dived to depths of 2,000 m and more, and scientists were able to film and collect marine life. Mechanical arms made it possible to perform experiments, and accurate navigation systems permitted returns to remote sites in the ocean. A number of smaller submarines allowed longer-term observation of depths of 300 m and less. One of the more whimsical submersibles was used in a marine station near Nice, France. The steel hull was connected to the surface by an air hose, and the investigator sat inside on a bicycle seat in a very cramped space. The first recorded observations in the Bay of Villefranche included one of a soup can on the murky bottom. Recently, researchers have used more modern submarines in the same area to observe spectacular bioluminescent planktonic jellyfish. To expand greatly the efficiency of deep-sea observation, remotely operated vehicles have been developed. These vehicles are unmanned but can make precise surveys and even take samples. Remotely operating vehicles are tethered to a ship by a cable, but a great deal of data are now collected by autonomous underwater vehicles, which are robots not connected to the ship. An interesting variant of AUVs are gliders, which use simple balancing devices to allow the vehicle to rise and fall through the water column or be moved by vanes in a constant direction by wave action. Ensembles of gliders now are being used by various shore-based laboratories and are deployed from ships because they are much cheaper than ship-based sampling. Nothing in shallow water, however, has matched the importance of scuba diving, developed in the 1940s. This form of underwater exploration was not used often or effectively until the late 1950s, when biologist Thomas Goro pioneered the study of coral reefs. Today, direct observations and experiments can be done on rich shallow-water marine biota. Although many advances have been made in diving and other technologies, the coming decades will see enormous strides toward remote sensing of the sea by satellite imaging. In the 1970s and 1980s, an American satellite known as the Coastal Zone Color Scanner provided images and conducted sophisticated light-based estimates of water temperature, chlorophyll, and other parameters. Now, new satellites are investigating with far more resolution. In conjunction with the new detectors, marine biologists are trying to use ground truthing to produce equations that relate color information received by satellites to measurements taken at sea. In the long run, this will allow us to process worldwide datasets, a capability that is crucial in our current studies of global climate change. The most recent advances in ocean observatories have taken advantage of global positioning system located fiber optic cable systems, with ports for remote video observation, sensing of physical variables such as temperature and current speed, and chemical measurements. This exciting new area is only now being developed and will allow a series of permanent and continuous observation posts to be established within shore and estuary locales, but also on the deep-sea floor and in mid-water locations. Most exciting is the Monterey Accelerated Research System in Monterey Bay, California, where a submarine canyon cuts the continental shelf and extends to the deep-sea floor. This cable has data collection ports, including video, and allows scientists to continuously monitor and observe remote localities with internet communication for research and education. The U.S. National Science Foundation has initiated a large-scale ocean observatories initiative, which combines observations from moorings, autonomous vehicles, and underwater cabled observatories. Observation and hypothesis testing. Marine biologists, like all scientists, use the scientific method, which is a systematic means of reasoning and observation. Marine biology, like all science, depends on a generalized system of observation and inference of the natural world known as the scientific method. This may sound unduly stiff and distant, but the scientific method is merely a systematic way to reason about and observe our world and universe. It depends on observations, deduction, and prediction. We are constantly making observations about the natural world, and many of these are repeatable. For example, we might find that all fish we observe live only in water. This would lead to a conclusion about the biology of fishes. Fishes live in water. The accumulation of specific observations to make a generalization is called induction. By contrast, we might take some premise and use logic to make a prediction. Such an inference, predicated on logical associations of conclusions with facts and premises, is a deduction. If you counted all the spectators in a football stadium drinking a beer, you might come to the conclusion that at 2 p.m. during the game, 10% of the spectators drink beer. That is an induction. Instead, you might reason that most spectators like beer, but all could not be drinking all the time because the lines at the beer concession are very long and only one beer is sold at a time. Therefore, you might deduce that only a fraction of the crowd will be drinking. If you knew the length of the game, how long it took to buy and drink a beer, how much blood alcohol it took to get drunk, and how fast alcohol is metabolized to non-inebriating products, you might be able to deduce how many spectators were holding a beer at any one time and how many are drunk. This line of reasoning is far more valuable because it has led you to develop a prediction that could be applied to other stadiums. Deduction has the beautiful property of prediction. Here's a more biological example. We might find that there is genetic variation in a population and, knowing that the environment may change, we might predict that some variants will perform better and become more frequent in the population as the environment changes. If we know how genetic transmission works and the relative survival and reproduction rate of those variants, we can predict the rate at which they will increase in frequency in the population. This is Darwin's theory of natural selection, which uses the method of deduction. This form of inference always depends on general premises and a logical pattern of reasoning to draw some specific conclusion. Most scientists strive to develop generalizations and theories from which predictions follow by deduction. Perhaps it is worthwhile to count all the days of the year that are cloudy and then conclude that most of the year is cloudy, but it would be much better to have a set of premises, a predictive relationship, and a theory to understand why it is cloudy most of the year. Induction, however, is a necessary part of science and can even be an inspiration for deduction. Most marine biological research requires extensive observations and correlations, but experimentation is usually the most efficient way to answer a question. Marine biological research involves a great deal of observation. In some cases, the observation is general and not directed toward any specific research problem. It is essential, for example, to know the distribution of temperature, salt content, water depth, and other properties of seawater because such information is required to solve a diverse array of specific problems. In other cases, observations are targeted toward more specific questions. To understand the migration route of a species of fish, it may be necessary to sample the ocean to detect tagged fish by remote signal or by catching fish directly at various times of the year and at various geographic locations and water depths. In many instances, observations by themselves cannot solve a marine biological problem. One of the most common types of observation is a correlation, which is an observed relationship between one factor and another. You might discover an increase of fish abundance with increasing water depth. This would be a positive correlation between abundance and water depth because both variables change in the same direction. On the other hand, you might discover a negative correlation, which in this case would mean that fish abundance decreases with increasing depth. As one variable decreases, the other increases. In either case, however, finding such a correlation does not prove that depth specifically is the cause of changes in fish abundance. The negative correlation might be coincidental. A decrease of fish abundance with increasing depth might be due to an increase of predator abundance with increasing depth. The next year, the number of predators might be in a different relationship with depth. This underscores a familiar saying among scientists: correlation does not prove causality. Experimentation is a much sharper and more powerful way of establishing cause. Suppose that, after finding a negative correlation between fish abundance and depth, you could perform an experiment and remove all the predators that are living in deeper water. If the prey fish then spread equally to all depths, you could reasonably conclude that the presence of predators, and not water depth itself, was the cause of the negative correlation. Experimentation is an important tool for both laboratory and field studies. Unfortunately, many marine problems cannot be approached by experimentation. Often, organisms and environments cannot be studied except by observation. This is especially true when the spatial scale is so great that it is impractical to perform experiments. Try to imagine changing the circulation of an ocean experimentally to study nutrient transfer, and you'll get the idea. It is possible, however, to formulate hypotheses that employ tests using distributional data. Marine biological research involves the testing of hypotheses and may involve experimentation or sampling. When solving problems in marine biology, additional observations beyond a certain point are not necessarily helpful. You could count all the fish in the ocean and still not know why they are abundant in some places but absent in others. To solve a scientific problem in a satisfying way, a hypothesis must first be stated. A hypothesis is a statement that can be tested. The following are examples of hypotheses. Predatory snails reduce the population size of mussels on the intertidal rocks on the coast of Monterey, California. Increasing temperature increases the rate of oxygen consumption of crabs. The following is not a hypothesis. Mermaids can never be observed, but they exist. I hope you can see the difference easily. One can test a hypothesis. To test a hypothesis, it must be possible to produce an outcome that shows the hypothesis is false. One makes a prediction, which must follow from the hypothesis. We therefore formulate an experiment, whose outcome will be consistent or not consistent with the hypothesis. If one has hypothesized that predators control a population, it is appropriate to remove the predator population and observe whether the prey population increases, as would be expected from the hypothesis. It is also possible that the premises of the hypothesis are inappropriate. Take the following hypothesis: sea stars cannot attack mussels, and they therefore have no effect on mussel populations. Because the premise of the first clause is incorrect, the hypothesis is inappropriate. All hypotheses should be internally consistent and testable, and they should be based on correct premises. Although some hypotheses are best tested by experiments, many cannot be. Sometimes the predictions will then be stated in terms of relationships. Even if the relationships could, on occasion, conceivably have more than one explanation. For example, we might state the following hypothesis: when circulation of a very large water body deeper than 100 m has a current speed of less than 2 cm/s, the oxygen there will decrease faster than it is replenished by circulation from shallow water, and the deep water body will lack oxygen. We obviously cannot perform an experiment on such a deep water body. We might then look at current speeds in all water bodies and classify on the basis of current speed those that lack oxygen. If the results of the classification are consistent with the hypothesis, we might look for any alternative hypotheses that could explain the same information. If none are obvious, then we might lean toward the correlation study as a correlation-based test of the hypothesis. Hypothesis testing is most powerful when specific predictions for an experimental treatment can be contrasted with difference from a control. The most difficult aspect of hypothesis testing is to formulate a hypothesis that lends itself to a specific program of experimentation or data collection. Figure 1.9 shows a technique that captures the best-known way to think deductively and formulate hypotheses. All science usually derives from initial observations that arouse curiosity, such as finding that a barnacle species is abundant only on the high shore. Then an explanatory model is formulated, which uses general principles to attempt to explain why that barnacle would be associated with a high shore location. One might argue that predation, which occurs only on the lower parts of the shore, prevents barnacles from surviving there, but the absence of predation on the high shore allows barnacles to settle as planktonic larvae and accumulate in this upper microhabitat. Now the crucial point arrives. One must formulate a hypothesis that is testable. The specific explanatory hypothesis here is that predation is more intense on the lower shore. Figure 1.10 shows a rocky shore on the outer coast of British Columbia, and you should note the great abundance of starfish below the mussel bed. Starfish can kill and digest mussels and barnacles, but they need to move about on very delicate tube feet, which work only in a moist environment. They are therefore able to rise onto the mussel bed only as the tide rises. Hence, we might expect that predatory starfish will have only the time to seize and eat prey on the lower shore because of the sluggish movement of the starfish. But how does one test that there is an effective shore height on predation? We could place a wire mesh cage over the rocks that keeps predators out but allows the rocky shore animals kept within the cage to function normally. A cage is placed on both the lower and upper shore and compared with open uncaged areas at the two levels. So the caged areas exclude predation, and the open areas are controls. The working hypothesis states that starfish would be able to kill mussels only on the lower shore, so we expect prey abundances to be much lower in the uncaged area on the lower shore but higher in the open area of the upper shore and also higher within cages at both lower and upper shore levels. The null hypothesis in this case would state, after the cages have been in place for a set period of time, there would be no differences in mussel population density between the open and caged rocky shore regardless of shore level. One must always remember that even an experimental result is a correlation of outcome with experimental treatment. The treatment effect, however, may have nothing to do with the hypothetical effect being studied. For example, the open low-shore area might show a decline because of full exposure to the sun, while higher-shore mussels have adjusted to this exposure. This may seem contrived, but it is consistent with the caging comparisons. In any test of a hypothesis, one must be aware of variation. Statistics is the field that deals with the calculation of trends and differences from repeated collections of information and assessments of variation. The difference in barnacle abundance between treatment and controlled may differ, but is the difference important? Two issues must be settled. First, a test of statistical significance must be established to determine whether the average barnacle density is greater within cages than outside cages. We need an estimate of variation and therefore need replicates of each treatment. If the variation among replicates is relatively low and the magnitude of mean difference high, then the difference may be statistically significant. Usually, a test is devised that can estimate the probability that the data are distributed non-randomly and to estimate the effect size of the factor you are studying. It is crucial to realize that the probability levels used in a statistical test are a reflection of nothing more than the probability of the data you collected and analyzed for this particular experiment, not a general truth of a scientific relationship. Habitats and life habits, some definitions. Some terms are necessary to describe life habits of marine organisms: Neuston, plankton; Nekton, benthos. It is useful to classify marine organisms by their general habitat. Plankton are organisms that live suspended in the water. They may have some locomotory power but not enough to counteract major ocean currents or turbulence. They include protists, animals, plants, and bacteria that are at most a few centimeters long. Neuston are organisms associated with the sea surface and include microorganisms that are bound to the surface slick of the sea. Nekton are usually larger animals that swim in the water column, but they can move against a current or through turbulent water. They range from small shrimp, crabs, and fish to the largest of whales. Benthos include animals and plants associated with the seafloor. Some animals are infaunal, which means they can burrow within the soft seabed, whereas others live on the seabed surface or are epifaunal. Most clams are infaunal, whereas oysters and barnacles are epifaunal. Mobile organisms associated with the seabed that can swim are said to be demersal. Figure 1.13 gives a general classification for marine habitats based on water depth. The intertidal zone is the range of depths between the highest and lowest extent of the tides. In some parts of the world, there is little or no tide, and wind mainly determines the vertical range of this fringing environment. The subtidal zone is the entire remainder of the seabed from the low water tide mark to the greatest depth of the ocean. Continental shelf habitats include all seafloor and open water habitats between the high water mark and the edge of the continental shelf. Seaward of the shelf is a series of oceanic or pelagic habitats. The epipelagic zone includes the upper 200 m of water, the mesopelagic zone ranges from 200 to 1000 m depth, the bathypelagic zone ranges from 1000 to 4000 m depth, and the abyssopelagic zone ranges from 4000 to 6000 m depth. Bathyl-benthic bottoms range from 1000 to 4000 m depth, and abyssobenthic bottoms range from 4000 to 6000 m depth. Hadal environments include those of the seabed and the waters at the bottoms of the trenches, often far deeper than 6,000 m depth. For example, the Marianas Trench reaches about 11,000 m depth. Chapter: The Oceanic Environment. The open oceans and marginal seas, the world's oceans, can be divided into open oceans and marginal seas. The ocean covers about 71% of the Earth's surface, and the southern hemisphere is more dominated by ocean than the northern hemisphere. The Pacific Ocean is the largest ocean and is less affected than the Atlantic by regional differences in climate or by river input. Island chains are most numerous in the Pacific, and volcanic activity is pronounced around its margins. Think of Mount St. Helens in Washington, Mount Fuji in Japan, and Krakatoa in the Southwest Pacific, and you'll soon understand why the rim of the Pacific is often called a ring of fire. The Atlantic Ocean is relatively narrow and is bordered by large marginal seas, example Gulf of Mexico, Mediterranean Sea, Baltic Sea, and North Sea. It drains many of the world's largest rivers. The Atlantic Ocean is affected to a larger degree by terrestrial climate and riverborne inputs of dissolved and particulate substances. The Antarctic Ocean has a solely water border with other oceans. Its northern boundary is the subtropical convergence, where colder, more saline water descends northward. A general pattern of west winds in the range of 40 degrees, 60 degrees south generates a surface current circling eastward around Antarctica. This and other important surface currents are illustrated in Figure 2. 1. Most marginal seas have unique oceanographic characteristics owing to their restricted connections with the open ocean and their usually shallow water depths. The restrictions allow local climate to strongly influence the marginal sea. For example, a shallow water barrier, or sill, restricts exchange between the Mediterranean and the Atlantic Ocean, and a local excess of evaporation relative to precipitation increases the salinity within the Mediterranean. 5 to 6 million years ago, a global lowering of sea level severed the connection with the Atlantic, and extensive evaporation led to the formation of large-scale salt deposits. Most other marginal seas have had histories of strongly changing conditions. Topography and structure of the ocean floor. The oceans share four main topographic features: the continental shelf, continental slope, the deep sea floor, and oceanic ridge systems. The continental shelf, a low-sloping platform, extends from the shoreline to roughly 10 kilometers to over 300 kilometers out to sea. Seaward of the shelf slope break. The continental slope increases in grade to about 120. The continental slope is usually dissected by submarine canyons that act as channels for downslope transport of sediment. The foot of the slope merges with the more gently sloping continental rise, which descends 2-4 kilometers to the abyssal plain, which averages 4,000 m depth. In some parts of the ocean, trenches in the seafloor occur just seaward of the base of the continental slope and may be more than 10,000 m deep. The trenches are long and narrow in map view and may run parallel to the shoreline. Isolated mid-oceanic islands may rise from the deep sea floor to the surface. In contrast, continental islands are located on the continental shelf. The oceanic ridges are linear features that rise 2,000, 4,000 m from the ocean floor and may reach the sea surface, forming emergent islands. The longest of the ridge systems runs the length of the middle of the North and South Atlantic Oceans, snakes around the southern tip of Africa, and runs northeastward into the Indian Ocean. Ridges are volcanic in origin and are often cut by transverse faults, or breaks in the Earth's crust. Rift valleys, at a ridge system's center, are parallel to the line of the ridge. The deep ocean basin floor consists of volcanic rock blanketed by soft deep-sea sediment. The sediment contains varying combinations of mineralized plankton skeletons, clay, and other minerals deposited from continental sources, volcanic rocks, and precipitates, such as manganese nodules, which are scattered on the seabed in certain areas. The skeletons of many organisms are composed of calcium carbonate, the material that makes up chalk. Many open ocean sediments are formed from a drizzle of calcium carbonate skeletons originating near the surface and make up, for example, foraminifera ooze. Calcium carbonate dissolves at great depth, however, and deep ocean sediments become progressively dominated by clays and, at certain sites, a radiolarian ooze of silica skeletons. Beneath the soft sediment covering and throughout the world lies the oceanic crust, made of sedimentary rocks and an underlying layer of volcanic rock. The Earth's crust is in continual motion, and the main topographic features of the ocean reflect this motion. The ocean floor can be divided into large sections, called plates, whose boundaries are ridges, trenches, or large transverse faults. Many geologists believed that some large-scale process caused the overall features of the oceanic crust. In the 1960s, scientists discovered that several features of rocks and sediments occurred in bands that were parallel to the oceanic ridges. As rocks crystallize, iron-rich magnetic minerals align to the Earth's magnetic field at the time of formation. Therefore, minerals that are forming now or in the very recent past have the magnetic polarity of the present-day Earth. Nearest a given ridge, the magnetic polarity indicated by the oceanic crust matches that of Earth's present magnetic field. In the next band on either side of the ridge, however, the magnetic polarity is reversed. The next bands are reversed again, and so on. The pattern exists for two important reasons. First, volcanic oceanic crustal rock is forming at the ridges and the rock is moving away from the ridges. But the Earth's magnetic field has reversed periodically over time, with the south magnetic pole flipping to north or the reverse. For example, before 780,000 years ago, the Earth's magnetic field was reversed relative to the present day. Thus, rocks formed at that time have magnetic minerals that crystallized with a magnetic orientation that is the reverse of minerals that are forming today or since 780,000 years ago. The mirror image of bands of rocks of alternating magnetic polarity on either side of a ridge was the key to understanding a major process called seafloor spreading. The oceanic crust, formed by volcanism at the ridge, was subsequently displaced from the ridge in both directions. This could happen only if new crust was formed through volcanic activity at the oceanic ridges and carried horizontally, as if on a conveyor belt, away from the ridges. Because the bands could be dated by means of radioactive minerals and the width of the bands was known, it was possible to calculate a likely range of speed of seafloor spreading, 25 centimeters y. It was later discovered that crustal material is dragged downward at trenches and melted into the upper mantle, the layer of the Earth beneath the crust. These sites of downward crustal motion are known as subduction zones. The great depth of the trenches reflects the downward dragging of the crust. The mechanism of seafloor spreading is a convective process occurring within the mantle. Heat is generated within the mantle, probably mainly by radioactive decay. The heat melts rock in the upper mantle, and volcanism in the mid-oceanic ridges is the result. Earthquakes are a direct cause of this movement, which often involves moving plates locked against each other for a time, or jerky lockup and movement along giant faults in the Earth's rock crust, with catastrophic release. Many of the world's most dangerous earthquake zones are at boundaries such as the San Andreas Fault in California, whose movement has caused many serious earthquakes. Earthquakes are also generated in areas of subduction, which nearly ring the Pacific Ocean. As an example, a very strong subduction zone exists in a region of the west coast of North America from northern California to southern British Columbia, Canada. Very strong evidence supports a history of very strong earthquakes about every 250 years in the Pacific Northwest of the United States. The last known earthquake that occurred in this northwest subduction zone, called the Cascadia subduction zone, in the year 1700, sent a tidal wave, or tsunami, toward the coast of the United States probably in 15 minutes, but also sent an enormous wave in the opposite direction across the Pacific Ocean to Japan in about 10 hours. Historical records of this tsunami allowed the accurate dating of the powerful earthquake in the Pacific Northwest. Seafloor spreading explains many of the Earth's topographic features. The continents consist of relatively low-density crustal material lying above a denser oceanic crustal layer. Continents move along with the spreading crust. The continental shelf is merely an extension of the continent below water. If you consider time spans of millions of years, the rate of seafloor spreading is significant. For example, 100 million years before the present day, the Atlantic Ocean was probably less than 2,000 kilometers wide, and South America and Africa were fairly close together. If the continental shelves are included, those two continents are found to fit quite well together, as pieces of a giant jigsaw puzzle. These two landmasses were probably once part of a supercontinent that divided, eventually producing today's continents. Seafloor spreading explains such continental drift. Throughout geological time, continents have had radically different arrangements, and even the major surface current systems have differed strongly from today's arrangement. The position of ancient continental blocks can be reconstructed using the orientations of polarity of magnetic minerals of ancient rocks. When the magnetic minerals crystallized, they pointed in the directions of the magnetic poles at the time of crystallization. It is therefore possible with the aid of computer-assisted algorithms to reconstruct where a continental block might have been, assuming you know the rock's age and orientation in space when the mineral crystallized. The origin of ocean crustal rock at the ridges and destruction of the trenches, combined with seafloor spreading, creates a large-scale seascape that strongly affects marine organisms. For example, the east coast of North America has been steadily moving westward for tens of millions of years, leaving a coastline that is generally a location of sediment formation and soft shores. By contrast, the west coast of North America is along a major fault line, the San Andreas Fault, and geological activity and volcanism have produced a very long coastline dominated by rocky shores as opposed to the sandy and muddy shores of the east coast. Sea level has also been strongly affected throughout geological time by seafloor spreading because mantle convective processes have vertical as well as horizontal motion. Large parts of the seafloor flanking mid-oceanic ridges move downward during the process of seafloor spreading. Using a wide range of magnetic and geological evidence, it is possible to locate ancient seafloor spreading centers and the vertical range that oceanic crust has been drawn down as it spreads from ridge centers. Seafloor spreading can lead to long-term changes in sea level in various parts of the ocean. For example, because of North America's long-term passage over a sinking plate, sea level has steadily declined from a high in the Cretaceous period, 65 million years ago, to an estimate of 170 m lower at present. This long-term decline is comparable in magnitude to the fluctuations of sea level of about 120 m during the most recent Pleistocene glacial age because of expansions and retreats of continental glaciers. The ocean above the seabed seawater. Many of the ocean's major features are due to the unique set of chemical and physical properties of water, including its dissolving power, high specific heat, transparency, and heat of vaporization. General properties of seawater. The formula for water, HO, indicates the component atoms of hydrogen and oxygen. Water molecules are asymmetric in charge, with a net positive charge on the hydrogen sides of the molecule and a net negative charge on the oxygen side. This overall polarity causes fairly strong attractions, called hydrogen bonds, between molecules. These attractions allow water to be liquid at atmospheric pressure and typical temperatures on the Earth's surface. Charge asymmetry also enhances the ability of water molecules to combine with ions, or particles. As a result, water is an effective and versatile solvent, and the ocean thus contains a diversity of dissolved substances. Water, including typical open ocean seawater, also has a very high specific heat, it takes 1 calorie to raise the temperature of a gram of pure water by 1 degree Celsius. Relative to most other liquids, it takes a great deal of heat to change water temperature. Seawater can store large amounts of heat, and moving currents can transport large amounts of heat along with the water itself. Water has a high heat of vaporization, and large amounts of heat are absorbed when water vapor is formed by evaporation. As a result, the ocean has a very strong effect, through the atmosphere, on both oceanic and adjacent continental climates. It takes a great deal of heat to change the temperature of the ocean. The ocean thus tends to smooth out temperature variation, an effect that is especially noticeable on coastlines that receive oceanic breezes. Temperature and salinity. Seawater temperature is regulated primarily by solar energy input and the mixing of other water. Seawater temperature is measured by means of mercury thermometers, thermistors, and satellite detectors. Thermometers have been used since their invention in the 18th century. Since the 1980s, semiconductor devices called thermistors have been used to directly record temperature and have an accuracy of 0.001 degrees Celsius. These are often installed on ocean buoys and can either transmit data by radio or record data that can be retrieved periodically. Finally, satellite radiometers are sensitive to infrared radiation and can measure sea surface temperature throughout the world ocean. At low latitudes, there is a net capture by the Earth of heat from solar energy, but at higher latitudes, the Earth tends to lose heat. As a result, there is a latitudinal temperature gradient of surface seawater. Surface transport and deep transport of heat energy by means of water currents are extensive, however, and the high heat capacity of water permits large transfers of heat among latitudes. Nevertheless, polar surface water temperatures hover at about 0 degrees Celsius year-round, whereas tropical surface water temperatures are almost constantly above 25 degrees Celsius. Local restriction of circulation causes further increases of temperature, sometimes approaching 35 degrees Celsius and higher in restricted lagoons and pools. Seasonal temperature changes are maximal in the mid-latitudes. Although temperature in high latitudes is low, the seasonal variation is minimal. Warm and seasonally constant temperatures are typical of the tropics. Seasonal change in the ocean is far more pronounced in the North Atlantic than in other oceans owing to the strong influence of continentally derived weather systems. In coastal North Carolina, temperature ranges over the year from roughly 3 degrees to 30 degrees Celsius. By contrast, the ocean-moderated Pacific climate has only an annual range of less than 5 degrees Celsius along the coast of California, and summer maximum temperatures are far lower than at comparable latitudes in the Atlantic. Bodies of water with restricted circulation and shallow depth, such as the Mediterranean, have greater annual temperature ranges than does the adjacent open ocean. Salinity is a measure of the dissolved inorganic solids in seawater. The salinity of seawater is the number of grams of dissolved inorganic solids per 1,000 grams of seawater. It is expressed as parts per thousand. Many of the elements in seawater are found in nearly constant ratios throughout the world ocean, even if the total salinity varies from place to place. The constancy becomes more pronounced with the increasing residence time of most elements in seawater. Residence time is the average time that a unit weight of a substance spends in the ocean before it is lost to sediments or to the continents. In the ocean, the elements found to be in constant proportions have a residence time on the order of 1 million years or more, as opposed to the mixing time of seawater, which is on the order of thousands of years. The relatively short mixing time homogenizes the various elements relative to each other throughout the ocean relative to the very long time they spend in the ocean before leaving. Other elements such as nitrogen have very short residence times, which are rapidly taken up and released by biological processes and vary greatly in proportion from place to place. Seawater is a complex solution. The solutes dissolved in it consist of both dissolved inorganic and organic matter, including dissolved gases. In addition, particles are suspended in seawater. The particles may be inorganic mineral grains, aggregates of organic particles, or living plankton. Dissolved inorganic matter enters the ocean mainly through river flow but also through atmospheric precipitation. The major elements are defined as those present in concentrations greater than 100 parts per million. These include chlorine, sodium, magnesium, sulfur, calcium, and potassium. By far, the dominant elements are chlorine and sodium, and indeed, when seawater evaporates, the salt sodium chloride is a very prominent residue. Minor elements are present at concentrations between 1 and 100 ppm. These include bromine, carbon, strontium, boron, silicon, and fluorine. Trace elements, those present at less than 1 ppm, include nitrogen, phosphorus, and iron. A large number of elements occur in minute quantities, in concentrations of parts per billion. The ratio of chlorine to other elements in seawater is essentially constant, despite overall changes of salinity. This fact enables you to estimate total salinity by measuring the chlorine content of seawater. Chlorinity is the total concentration of chloride per 1,000 milliliters of seawater. Salinity is approximately 1.81 times the chlorinity. A simple chemical titration of chloride ions from seawater can therefore be used to estimate total salinity. Because a saline solution conducts electricity, conductivity can also estimate salinity, but corrections must be made for temperature. Although it is accurate only to about 2 ppt, a simple optical refractometer can also measure salinity because increased salt content increases light refraction, the degree to which water bends light. In most oceanographic applications, you will find that salinity is not measured in terms of dissolved inorganic solids. Although the ratios of major elements with long residence times are similar throughout the ocean, the slight differences make measures based on total dissolved solids inconsistent. Oceanographers therefore generally express salinity using the practical salinity unit, which is a measure of the electrical conductivity of the seawater relative to a standard potassium chloride solution at 1 atmosphere pressure and 15 degrees Celsius temperature. Practically speaking, the numerical units between PSU and solute-based salinity measures are nearly identical. Today, most marine biologists measure salinity by means of a relatively inexpensive device that measures conductivity. In terms of practical salinity units, salinity is expressed with no qualifier. Therefore, a salinity of 30 on the practical salinity scale is described as 30. In the open ocean, salinity ranges from roughly 32 to 37. Locally, salinity is regulated by a balance of dilution and concentration processes. Latitudinal variation in the balance of precipitation and evaporation causes slight salinity maxima at 30 degrees north and S latitude, with a minimum at the equator and declining salinities at higher latitudes. Most marginal seas differ from the adjacent open ocean owing to their restricted circulation and local factors such as excesses in rainfall, river input, or evaporation. The Baltic Sea, located in northern Europe between Sweden, Finland, and continental Europe, has an excess of river runoff and restricted circulation with the Atlantic. As a result, the salinity of much of the inner Baltic is 5 or less. By contrast, the Mediterranean has an excess of evaporation relative to precipitation and runoff, and its salinity is usually greater than 36, which influences the adjacent North Atlantic Ocean. Oxygen in the sea. Oxygen is added to seawater by mixing with the atmosphere and by photosynthesis. It is lost by respiration and by chemical oxidation of various compounds. Oxygen from the atmosphere dissolves in seawater at the sea surface. When the wind is strong, larger amounts of atmospheric oxygen are mixed into the surface waters, which may then be mixed with deeper waters. The amount of oxygen that can be dissolved into seawater decreases with increasing temperature and decreases slightly with increasing salinity. The balance of organism respiration and photosynthesis also affects the amount of dissolved oxygen in seawater. Photosynthesis is the light-driven process that plants use to manufacture carbohydrates. Oxygen is a byproduct of the process. During respiration, the opposite process occurs. Carbohydrates are oxidized to provide energy for the organism, and oxygen is consumed. Respiration losses can usually be accounted for in terms of bacterial decomposition of organic matter and by animal and plant respiration. An idealized equation for respiration is CH12O plus 6O6CO plus 6HO. Plus energy in photosynthesis. This reaction goes to the left, although the biochemical details are very different. Chemical oxidation of organic matter also may reduce dissolved oxygen in seawater. Restriction of vertical and horizontal water circulation often tips the balance toward low oxygen concentration. In some major water bodies, the supply of organic matter and its role in oxygen depletion outstrip the role played by the mixing of bottom waters of the basin with other oxygenated waters. For example, the bottom waters of many Norwegian fjords have restricted circulation with the open sea owing to the presence of a sill, or shallowing, at the entrance, which permits only surface water to leave the fjord. Decomposition of organic matter tends to consume the oxygen in the stagnant bottom waters. The vertical structure of seawater, with low-density seawater above and higher-density seawater below, may stabilize the water column and strongly influence local oxygen concentration. For example, particulate organic matter may accumulate in the thermocline, a zone of great increase of water density with depth, resulting in bacterial decomposition of the organic matter and consumption of oxygen. This results in the local absence of oxygen or oxygen minimum layers at depths of 200–1,200 m, which are especially common in the eastern North Pacific Ocean off the coast of California but are also found in the tropics of both the Atlantic and the Pacific and throughout the eastern Pacific. In estuaries, organic matter may accumulate in deeper quiet waters, resulting in lowered oxygen and sometimes mass mortality of fish and bottom organisms. Light environmental light originates mainly from the sun and is therefore strongest in surface waters. Light energy that reaches the earth emanates mainly from the sun, and therefore, most light enters the ocean from above at the surface. Light energy ranges from shorter wavelength ultraviolet light to light within the visible spectrum to light in the infrared and longer wavelength spectrum. At high angles near midday, very little sunlight is reflected from the sea surface. Much of the light is either absorbed by seawater and particles or scattered by particles in the water. Light intensity declines exponentially with increasing depth, as shown in Figure 2.12. The ultraviolet and infrared parts of the spectrum are very strongly attenuated with depth, so only the visible part of the spectrum reaches any appreciable depth. Blue light penetrates better than red light. Because it is the energy source for photosynthesis, light is crucial for life. Most marine animals also depend on light, so they can move about, detect prey, and spot predators. Light intensity near the surface can be sufficient to depress biological activity through the deactivation of protein and DNA. Ultraviolet light is damaging, especially in the clear waters of the tropics, where significant amounts will penetrate to depths of 10 m. Some intertidal plants use calcium carbonate to reduce damage by absorbing the ultraviolet light. Some corals have pigments that absorb ultraviolet light. In more turbid waters, ultraviolet light is no real problem, and well over 90% of it is absorbed in the top meter of water. The effect of sunlight diminishes greatly with depth. In turbid shelf waters, light is strongly limiting to photosynthesis even at depths of 10 m. At depths of 30–50 m, visibility is greatly reduced. This is especially true for color discrimination, and animals living in deep water do not have especially good color vision. In the open sea, at depths much greater than 1000 m, there is no detectable sunlight. The animals in the deep oceanic world see only a faint glimmer from above, if anything at all. Circulation in the open sea, patterns and causes surface circulation. Surface oceanic currents are controlled by the interaction of the planetary wind system and the Earth's rotation. Two factors interact to produce oceanic surface currents: the planetary wind system and the Earth's rotation. More sunlight is captured near the equator than near the poles, and the heated air tends to rise and move to higher latitudes and then sink, with more such vertical cells successively created at higher latitudes. This overall air circulation system drives the planetary winds, which drag over water and generate surface currents. The Earth rotates from west to east, which deflects the air as it rises and sinks. This modulates the direction of the winds and surface currents. Let's consider the factors that interact to produce surface water current circulation patterns. The Coriolis effect and surface circulation patterns the Earth's rotation causes. A deflection in surface current direction called the Coriolis effect, which affects water flow on many geographic scales. The Earth rotates once a day on its axis. A particle attached to the Earth's surface at the equator must travel from west to east more rapidly than one attached near the pole because the particle at the equator must traverse a longer distance in each day's rotation. If a parcel of water not attached to the Earth moves toward the north in the northern hemisphere, it is moving to a location of lower eastward velocity. Because the northward moving parcel of water has greater eastward momentum than the water into which it is moving, it will have a relative deflection toward the east. That deflection is toward the right. If the parcel is moving south, it moves from an area of lower eastward velocity into a region of increased eastward velocity. Relative to the local regime, the water will lag behind and will deflect toward the west. Once again, that deflection is toward the right. The effect of the Earth's rotation on moving air and water is known as the Coriolis effect. It causes a deflection to the right for water traveling in the northern hemisphere. It causes a deflection to the left for water traveling in the southern hemisphere. The Coriolis deflection works in the same direction. If you are traveling east or west, let us consider the northern hemisphere. If you are traveling toward the east, you will be moving in the same direction as the eastward rotation of the Earth. The centrifugal force tends to make you fly away, perpendicular to the Earth's axis, but there is a strong component of motion directed toward the equator, which makes the object turn to the right. As an object is moved toward the west, its motion is moving westward perpendicular to the Earth's axis, but its tangential velocity is decreased because the Earth beneath is moving in the opposite direction. Gravity tends to make the particle move to the pole or to the north in the northern hemisphere. So rightward movement also occurs in the northern hemisphere for eastern and western motion, and the Coriolis movement is again toward the left for eastern or western motion in the southern hemisphere. The major oceanic surface currents are determined by the planetary wind systems modified by the Coriolis effect. The most conspicuous feature of the large-scale ocean surface is the presence of surface currents, which are driven by the planetary wind system. The winds are caused by the rise of air heated by the sun and the sinking of the air as it cools. These movements are influenced by the Earth's rotation. In the latitudinal band of 30 degrees, 60 degrees north and s, the westerlies move air toward the northeast or the southeast. Between the equator and 30 degrees north or between the equator and 30 degrees south latitude, the trade winds blow toward the southwest or the northwest. These two wind systems, together with the direct action of the Coriolis effect, move tremendous volumes of surface water in large circular patterns known as gyres centered around 30 degrees latitude N or S. These gyres move clockwise in the northern hemisphere and counterclockwise in the southern hemisphere. In the North Atlantic, the westerlies push water toward the northeast, but the Coriolis effect results in eastward flow. Farther south, the trade winds drive water toward the southwest, but the Coriolis effect produces westward flow. The constraining shapes of the continents in the North Atlantic contribute to the clockwise flow of the North Atlantic gyre. In summer, persistent winds tend to reinforce these patterns. On the east coast, the clockwise flow brings warm water from the south to the New England region. In contrast, the clockwise Pacific flow brings cold water from higher latitudes to the coast of central California. The Coriolis effect deflects water moving under the dragging force of the wind. The wind causes the water to move in surface sheets. Because of friction, these sheets drag on layers beneath. In the northern hemisphere, the Coriolis effect causes each successive sheet to deflect to the right relative to the sheet above. Theoretically, this should produce a spiral circulation with increasing depth. In practice, the net direction of surface water is approximately 90 degrees to the right of the wind in the northern hemisphere and 90 degrees to the left of the wind in the southern hemisphere. This motion is known as Ekman transport. Western ocean boundary currents are caused by winds combined with the Earth's rotation and result in large-scale transport of surface water from the tropics to higher latitudes. The oceanic boundary currents on the western sides of the major Atlantic and Pacific gyres are famous for their speed and ability to transport large amounts of water. These currents are well defined with strong thermal boundaries between the current and the surrounding oceanic water. The Gulf Stream, for example, originates from within the Gulf of Mexico, flows northward along the eastern coast of the United States, and moves eastward in a more diffuse current across the Atlantic toward Ireland and Great Britain. Off the coast of the southeastern United States, current speeds can be as high as 2 ms, down to depths of over 450 m. More than 50 million ms of warm water may be transported northward along the North American Atlantic coast. Large amounts of heat are stored in the current at its source, and this heat is transported northward, veers to the northeast, and is eventually lost when the waters reach the mid-North Atlantic, when the waters are now cold and sink. Westerly winds blowing across North Atlantic waters help ameliorate the climate of the British Isles, which would otherwise be colder. As a result, subtropical vegetation grows well in Ireland and southern Scotland. A similar situation exists in the western North Pacific Ocean. The Kuroshio Current brings warm water toward the north that then moves eastward and loses its heat in the North Pacific. The climate in the Pacific Northwest of the United States is strongly affected by westerly winds blowing from the Pacific toward the west coast of Oregon, Washington, and British Columbia. Eddies of meandering parts of boundary currents may form cold core and warm core rings. Western Ocean boundary currents are often very well defined, but at some point downstream, they become sluggish and may meander. This is especially true in summer when there is less difference between the temperature of the oceanic boundary current and the usually surrounding colder water. Occasional meanders of the Gulf Stream form loops, which enclose pockets of cold water. These are known as cold core rings. They are usually formed between the Gulf Stream and the continental slope area, mainly off the coast of Nova Scotia. They then tend to travel southward, only to dissipate a few months later. These rings often have unique assemblages of plankton and fish. Occasionally, a meander of the Gulf Stream will enclose a still warmer summer blob of water on the northeastern U.S. continental shelf and drift on the shelf toward the shore, forming warm core rings. Commonly, large numbers of tropical fish, such as butterflies, can be found in coastal waters of Long Island, New York. These fish originated in Florida waters, drifted northward in the Gulf Stream, and then drifted shoreward, usually in a warm core ring. Winds and the Coriolis effect combine to cause upwelling, which brings nutrient-rich, deeper water to the surface. The Coriolis effect strongly influences water movement on the continental shelves of the east sides of oceans. In the northern hemisphere, a wind blowing directly from the north will deflect surface waters to the right or to the west, owing to Ekman transport. In the southern hemisphere, a wind from the south causes a leftward deflection of surface water, also to the west. The upward movement of deeper waters near the coast compensates for these offshore surface water movements. This phenomenon, known as upwelling, brings nutrient-rich deeper waters to the sunlit surface and fuels large-scale phytoplankton blooms, which in turn support major fisheries such as the anchovy fishery off Peru. Periodic shutdowns in this upwelling system occur, causing major climate oscillations in the region around Peru, but also worldwide. We discuss these oscillations, known as ENSO, in Chapter 3. Extraordinarily strong surface water motion is generated by storms and earthquakes. Cyclonic storms are rather common in both the Atlantic and the Pacific and may last for several weeks. The overall storm systems may move at speeds of a few kilometers per hour and can cover tens of thousands of square kilometers. Wind velocities of 160 kilometers or more per hour are common, and wave surges of 10 m over coastal areas are possible. Hurricanes can completely destroy a coral reef and cause other terrible effects. In 2005, for example, Hurricane Katrina caused tremendous damage to the people of New Orleans when the levee system broke, and ocean surge and saltwater intrusions from Hurricane Rita in the same year caused major damage to low-lying coastal forests. Earthquakes or slumping of submarine sediment can generate waves known as tsunamis, which may travel at speeds in excess of 600 km/h. Their wave height may be very small in the open sea, but if a shallowing coastline causes the water to pile up, tremendous destruction can be caused by the resulting wave surge, which can exceed 5 m. In the late 19th century, tens of thousands of people on the coast of Japan were killed by one especially destructive tsunami. A December 2004 earthquake of magnitude 9.1-9.3 near Sumatra generated an enormous tsunami throughout the Indian Ocean, reaching heights of 30 m. More than 200,000 people perished, and the arriving wave caused a great deal of damage to some coral reefs in the eastern Indian Ocean. The 2011 magnitude 9.0 Tohoku earthquake off the northeast coast of Japan generated waves on shore up to 45 m, probably killing about 20,000 people and destroying much of the large Fukushima nuclear power plant complex. Small-scale changes in current systems, combined with latitudinal temperature change and coastal irregularities, may isolate adjacent coastal regions. Previously, we discussed the role of seafloor spreading in continental drift. Because of these processes in the past 50–100 million years, much of the world's coastlines consist of north-south coastlines. For example, the Pacific coast of North America stretches all the way from northern Alaska to the very extreme southern hemispheric tip of South America. These coastlines exist along a large latitudinal gradient of temperature and variation of current systems. Also along these very long coastlines, we see a large number of projections of land, such as Cape Cod, Cape Hatteras, and the tip of Florida as we move southward along the Atlantic coast of eastern North America. These projections are also breaks of sea temperature change and isolation of coastal populations because of separated sea current systems. These breaks may be isolating barriers to migration and might be limiting barriers because the temperature changes too much for species to extend past a given barrier, such as living north of Cape Cod where temperatures drop substantially relative to southerly waters. Seawater density and vertical ocean circulation. Seawater density, an important property affecting the vertical movement of water, is controlled mainly by salinity and temperature. The density of seawater is expressed as the number of grams per cubic centimeter. Density increases with decreasing temperature and increasing salinity. Seawater density is only a few percent greater than that of distilled water, usually ranging between 1.02 and 1.07 GCM. Strong depth gradients in water density occur at many scales in the ocean. Vertical gradients in seawater density are known as pycnoclines. Because water density is affected significantly by variation in temperature, solar warming should play an important role in the vertical thermal structure of the ocean. This can be seen even at the scale of centimeters if you dip your hands into a tidal pool on a hot day. The top few centimeters will be warm, but the water beneath will be considerably colder. This principle works on the larger scale of a coastal water body with a depth of 20–50 m, such as Long Island Sound, which lies between Long Island, New York, and Connecticut. In the summer, solar warming from above produces a warm layer approximately 3–10 m thick, which is mixed by the wind and is isothermal throughout its depth. Beneath this mixed layer, a zone of rapid decrease in temperature, the thermocline, ends in a colder, deeper layer of relatively constant temperature. Due to the effect of temperature on density, this arrangement is very stable because the low-density warm water rides above the high-density cool water. Wind therefore does not mix the layers very effectively. As autumn sets in, this stratification of density begins to break down, and winds more easily mix the layers until the winter decrease of temperature results in a well-mixed and cold isothermal water column from the surface to the seafloor. On the largest spatial scale, the tropical ocean contains a surface warm layer, a deeper thermocline, and a still deeper cold layer. At the surface, the sun provides heat year-round, and the deeper waters are kept cold by deep circulation. The precipitation-evaporation balance in the tropics tips toward precipitation, which further reduces the surface water density. Vertical circulation and deep water circulation are regulated primarily by differences in water density, generated at the surface usually at high latitudes, followed by sinking and travel to lower latitudes. In contrast to the major surface current features, deep oceanic circulation is characterized by movement of large water masses whose unique temperature and salinity characteristics are acquired at the sea surface at high latitudes. The high-latitude water masses sink owing to their high density and move to lower latitudes. Beneath the surface of the ocean, the water masses reach different levels, which are determined by the densities of the different water masses. These water masses are stacked like a layer cake in descending order of increasing water density. Figure 2.18 shows an idealized cross-section of the Atlantic Ocean. Note the continuity of deep water masses from the surface at high latitude to lower latitudes and greater depths. The vertical structure of the Atlantic is explained as follows. In the Weddell Sea of Antarctica, a higher density of colder water is enhanced by slightly increased salinity generated by sea ice formation. Thus, the very dense Antarctic bottom water is formed near the surface, sinks, and moves along the bottom toward the northern hemisphere. The North Atlantic deep water forms in the Norwegian Sea, but it is not as dense as the AABW. It sinks and flows southward, moving above the AABW. The Antarctic intermediate water is formed near the Antarctic Circle but is not quite as cold and saline as the NADW. It sinks but comes to rest just below a tropical surface layer of warm low-density water and above the NADW. These movements form a series of layers of progressively lower density water toward the sea surface. Surface waters from the Ross Sea, on the side of Antarctica opposite the Weddell Sea, sink and eventually form cold and saline deep ocean bottom waters at lower latitudes of the Pacific Ocean. Of course, there is considerable mixing at water mass boundaries, but the masses nevertheless can be traced by characteristic temperature-salinity combinations. The time of movement is on the order of hundreds of years. The origin of water masses is crucial for the maintenance of deep water life because the water originates at the surface, where it is saturated with dissolved oxygen. Consumption of oxygen during transport is insufficient to make the water masses anoxic, and the deep-sea bottom waters are therefore largely oxygenated. This allows deep-sea organisms to have an ample supply of oxygen, usually. Water exchange is limited in some deep-sea basins, and these are anoxic. For example, the Carayaco Trench is a 1,400 m deep zone off a broad shelf region north of Venezuela. Oxygen is absent within this trench, and all chemical processes are affected by the absence of oxygen. The Coriolis effect modifies deep circulation, and some large-scale sedimentary features show deposition that follows the expected deflection. One might intuitively expect deep ocean currents to be sluggish, and this can be true. There are, however, some notable exceptions. Earthquakes whose foci are beneath the continental slope often set off rapid downslope movements of a water-sediment mixture known as a turbidity current. These currents, which are of sufficient force to transport coarse sediment, were discovered by virtue of their ability to snap submarine communication cables. Even on the abyssal seabed, currents can be fast, on the order of 50, 60 centimeters s. The deep sea is thus not necessarily a monotonous and static environment. In some areas, large volumes of water are funneled through narrow openings in submarine ridge systems and through narrow openings between marginal seas and the open ocean. There, current velocities are much higher than those on the rest of the deep seafloor. The surface and deep ocean are connected by means of a global conveyor belt circulation. The surface currents, driven by the planetary winds, and the deeper currents, regulated by density, are coupled through the global conveyor belt system. We can see this by examining the surface origin in the North Atlantic of cold seawater as the Gulf Stream moves northeastward at high latitudes. Here, the surface water, being cold and dense, sinks and moves at depth toward the south, eventually connecting with some of the AABW and later moving northward into the Pacific and warming and rising toward the surface, mixing with surface waters. This surface flow moves through the Indian Ocean, around the southern tip of Africa, and back into the Atlantic. The flow patterns are slow and not well understood. It has been recently argued that global warming might shut down the origin of the sinking water in the North Atlantic and that this might in turn shut down the strength of the Gulf Stream. Thus far, there is little evidence that such a shift has happened, although physical oceanographers are actively monitoring water motion in the North Atlantic. The edge of the sea waves and the shoreline wind moving over the water surface sets up a series of wave patterns, which cause oscillatory water motion beneath. Wind that moves over the water surface produces a set of waves, which appear on the surface as a series of crests and troughs moving in the direction of the wind. This occurs even though no large current is in motion. During a storm, one first notices the appearance of a series of waves and then an increase in the height of the waves. Wave height is proportional to the wind velocity, the duration of the wind, and the fetch, or distance over which the wind acts. Waves are defined by their period, wavelength, wave height, and velocity as well as by the water depth. Although waves are not always symmetrical and evenly spaced, a few dimensions adequately define their geometry. Wave height h is the vertical distance of crests from troughs. Wavelength L is the distance between successive crests, troughs, or other specified points. Period T is the time of passage of successive crests past a reference point. Finally, velocity v is the speed at which a crest or other specified point travels. The variables v, l, and t are related by the equation v equals lt when waves are symmetrical, water particles move in orbits. The diameters of these orbits decrease with increasing water depth and become insignificant at depths greater than L2. As a wave approaches the shore, it becomes affected by the presence of the bottom when the depth is less than L2, then the wave eventually breaks. Surface waves set the water into orbital motion at depths of less than L2. When the water becomes shallow enough, the orbital motion occurs at the bottom, and the waves at the surface begin to feel the bottom or be affected by its presence. The geometry of the wave begins to change, and the wave height increases. These effects are due to the insufficient space available for complete orbits to occur. The water is effectively pushed upward as the wave approaches the shore. The orbital motion becomes more and more elliptical, and water and mobile bottom sediment are moved shoreward, sometimes with great force. When HL exceeds 1.7, the wave becomes unable to sustain its own weight and collapses, or breaks. Shorelines greatly alter the pattern of waves and currents. Waves often arrive with their crestline at an angle to the shore. This produces currents parallel to the shore known as longshore currents. Occasionally, longshore currents encounter an irregularity in the shoreline and suddenly run offshore in concentrated and quite dangerous rip currents. Longshore currents are responsible for extensive erosion and transport of beach sands along outer coast beaches. The effect often makes the dredging of channels into outer beach bars a hopeless task. Often, misguided beach engineers place groins on beaches to prevent erosion. This has the effect of choking off the sand supply farther down current, along the beach. As a result of these practices, many resort areas, most notably Miami Beach, Florida, have lost their bathing sand beaches in the past. Irregularly shaped coastlines can strongly affect the direction and speed of incoming waves. As a wave front approaches the shore, horizontal water velocity diminishes as the water becomes shallower. Because the parts of a wave arriving on either side of a headland are still in somewhat deeper water, they will travel faster than the part of the wave striking the peninsula. This results in the wave refracting about the peninsula. The degree of refraction may focus wave energy on the headland, and this accelerates the erosion of the headland relative to the straighter coastline adjacent to the peninsula. The coast may be soft sediment or outcrops of rock under active erosion. In quiet water, soft sediment accumulates as sand or mudflats, where sediment movement is minimal and the slope of the flats changes little throughout the year. These flats can be hundreds to thousands of meters wide, as on the northern coast of France or on the flats of the Bay of Fundy. On more exposed beaches, strong wave action causes extensive sediment transport and a seasonal change of the beach profile owing to winter storms. Figure 2.23 shows some features of an exposed beach. A series of wind-blown dunes, sometimes stabilized by vegetation, lies behind the beach. A relatively horizontal platform, the berm, extends to a break in slope. Owing to storms, the winter beach profile then increases strongly in slope to the low tide mark. Seaward, a complex of troughs and offshore bars develops. Often, emergent barrier islands develop that protect large lagoonal complexes. Rocky coasts develop where outcrops of rock occur in geologically youthful terrains. The topography of such coasts depends on the local rock type and wave action. Sandstones, for example, are weathered and eroded into sand particles, which may produce a sandy beach at the base of a rocky outcrop. By contrast, highly cemented sedimentary deposits and crystalline rocks maintain their hard surface, and hard rock shores develop, as seen often in coastal areas of Maine and Northern California. Tides' gravitational effects of the moon and sun, modulated by the Earth's rotation and basin shape, cause periodic tides. The force of gravity acting between any two bodies is proportional to the product of the masses of the two bodies and inversely proportional to the square of the distance between them. Both the sun and the moon exert significant gravitational attraction on the ocean, but the moon dominates the tides because it is closer to the Earth. Keep in mind that the Earth is rotating about an axis once per day, but the Earth is also rotating about the Sun with a period of about 365.26 Earth days. The Moon is rotating about the Earth counterclockwise with a period of about 27.32 Earth days. Tidal motion can be measured throughout the ocean, but it is especially noticeable at the shoreline in the form of tidal currents and vertical water motion. The extent of the tide is largely determined by the difference in gravitational attraction on either side of the Earth relative to the Moon and Sun. On the side closer to the Moon, which has more gravitational effect than the Sun, the gravitational attraction pulls water toward the Moon. On the opposite side of the Earth, a minimum of gravitational attraction combines with the Earth's spin to produce a net excess of centrifugal force. This creates a tidal bulge away from the Earth. Corresponding depressions will exist on parts of the Earth between the bulges, where there is no net excess of gravitational pull relative to centrifugal force. Because the Moon passes over any point on the Earth's surface every 24 hours and 50 minutes or once each tidal day, ideally there should be 2 low and 2 high tides per day. Because the Moon's position relative to the Earth's equator shifts from 28.5 degrees north to 28.5 degrees south, the relative heights of high and low water differ geographically owing to changing vectors of gravitational attraction. The Sun interacts with the Moon to cause variation in the vertical range of the tide. During times of full or new moon, when the Sun, Earth, and Moon are in line, the gravitational force exerted by the Sun amplifies that of the Moon, and maximal tidal range at spring tide is achieved. When the Sun, Earth, and Moon form a right angle at times of quarter moon, the gravitational effects tend to cancel each other out, and neap tide occurs, with the minimum vertical range. 2 spring tides and 2 neap tides occur each lunar month. Tidal periodicity has both a semi-diurnal component and a diurnal component. In semi-diurnal tides, one finds 2 approximately equal high tides and 2 approximately equal low tides each day. These cycles are characteristic of the east coast of the United States. At the other end of the spectrum, some coasts under some conditions are dominated by the diurnal tide component with a single high tide and a single low tide each day. Finally, mixed tides are also common, where the semi-diurnal and diurnal components combine to give 2 high and 2 low tides each day but with unequal ranges. On the west coast of North America, it is common to have 2 high tides and 2 low tides of different heights each day. Figure 2.25 shows some tidal patterns over several days under different tidal regimes, the presence of irregularly shaped basins, the tilt of the Earth's axis, and the Coriolis effect also cause significant deviations from the idealized expectation of 2 highs and 2 lows each day. Each basin develops standing oscillations that resonate with tidal forces. If the travel times of these oscillations are short relative to the period of the tide generating force, the timing of the tide will be like that expected for an uninterrupted sphere covered with water. If travel time is long relative to the tidal period, the timing of the tide will be out of step with that expected for such a surface. The Coriolis effect has a great influence on tidal heights and currents. When affected by the Earth's rotation, tidal currents produce in the center of seas and oceans rotating systems around amphidromic points, or points where no change in tidal height occurs over time. In the open sea, both vertical tidal range and the strength of tidal currents tend to be modest. In coastal bays, however, volumes of water tend to be forced into ever smaller blind bays, and as a result, currents and vertical tidal ranges are greater than on the adjacent open coasts. In areas with salt marshes, creeks tend to have higher tidal ranges than the adjacent open coasts. The most dramatic examples, however, are funnel-shaped basins such as the Gulf of California and the Bay of Fundy, where vertical tidal range exceeds 10 m. Tidal currents are also strong there, and a pronounced incoming tidal wave, or bore, can be seen, for example, on the tidal flats of the Bay of Fundy or in certain rivers such as the Yangtze in China. Usually, there is a great difference of timing of the tides of bays relative to the adjacent open coast. Tides can also be extremely slight given the proper conditions. Because of the various interference effects of landmasses, areas such as eastern Denmark, the Gulf Coast of the United States, and the Caribbean coast of Panama have almost no vertical tidal movement. Water levels in these places tend to be dominated by wind action. Estuaries are coastal bodies of water where water of the open sea mixes with fresh water from a river. An estuary is a partially enclosed coastal body of water that has a free connection with the open sea but whose water is diluted by fresh water derived from a source of land drainage, such as a river. On the large end of the spectrum, Chesapeake Bay. drains 5 major rivers and includes a complex set of creeks, bays, and main channels. On the small end of the spectrum are individual rivers whose salinity changes with every tide. Nearly all large estuaries have been strongly influenced by the rise in sea level since the last glacial maximum. Chesapeake Bay's present extent stems from the drowning of the Susquehanna and adjacent rivers. Many estuaries, especially those on the east and Gulf coasts of the United States, owe their general form to the rise of sea level and the formation of barrier bars, which enclose lagoons of relatively lower and more variable salinity than the open ocean. The Outer Banks of North Carolina are barrier bars that protect an inland water system of great biological richness. Water movement in estuaries depends on the amount of river discharge, tidal action, and basin shape. Some estuaries have remarkably predictable salinity gradients and circulation. Others are far less predictable, and salinity and circulation depend on strong variations in freshwater flow from the watershed and variable connections with the open sea. Overall, there is a basic estuarine flow. Relatively low-density river water flows downstream and eventually comes into contact with saline water mixing in from the coastal ocean. Owing to its lower density, the river water tends to rise above the denser saline water mass moving upstream along the bottom from the adjacent ocean. In a highly stratified estuary, the layers are quite separate. With moderate wind action and tidal motion, mixing occurs at all depths and vertical exchange occurs, causing the salinity of both the upper and lower layers to increase seaward. In such a moderately stratified estuary, a cross-section would show that the isohalines, or lines of equal salinity, are inclined upward toward the sea. In these estuaries, water tends to flow up the estuary at depth and down the estuary at the surface. Such a flow pattern is found in the lower part of the Hudson River and in Chesapeake Bay. The river slope and total water flow are important determining factors of salinity structure and tidal action. In fjords, a previous history of glacial scouring results in a long stretch of river, where the bottom is nearly horizontal. In these cases, tidal effects tend to cause water flow far up the estuary. In the Hudson River estuary, which has such a glacial history, water is elevated only about 1 m over a stretch of about 250 kilometers from New York City to a dam near Troy, New York. As a result, tidal action extends far upriver. By contrast, a greater slope and higher water flow cause far less tidal effect to extend northward into the Mississippi River. Vigorous tidal mixing in relatively small estuaries tends to homogenize the vertical salinity gradient and results in a vertically homogeneous estuary. Because of strong tidal control, the salinity at any point in the estuary changes radically depending on the state of the tide. At low tide, the salinity is dominated by downstream river flow, whereas at high tide, the inrush of seawater increases the salinity. This mixing can occur only in very shallow estuaries, such as the small freshwater rivers entering salt marshes. Most estuaries experience strong seasonal effects and receive water, dissolved substances, and particulate matter from tributaries and runoff in the larger watershed drained by the estuaries and freshwater rivers. In the late winter and early spring, storms and snowmelt can cause an increase in water flow known as the freshet. This increase often brings into the estuary nutrients that sustain the productivity of the estuarine food web. If spring flow is small relative to the total volume of water in the estuary, as is true in Chesapeake Bay, then lines of equal salinity move down with the spring freshet but only slightly. In the Hudson River, however, discharge varies a great deal, and salinity in the lower part of the estuary can change greatly seasonally and between years. Chapter Climate Oscillations and Climate Change. Climate change, oscillations, and long-term trends. Oscillations and directional change in climate occur at large oceanic spatial scales. They interact to result in interannual climate variability. Short-term weather systems such as hurricanes occur on the time scale of days to weeks, but many other fluctuations in weather occur on longer timescales, the ones we associate with properties of climate, which refers to weather properties that characterize a region over longer timescales such as several years to decades to centuries. This leads us to a discussion of major oscillations and their interaction with longer period trends in climate that might occur over decades or centuries. Multi-decadal oscillations in climate are a major pattern observed throughout the world's oceans. World climate patterns in recent decades are usually a combination of oscillations combined with a long-term warming trend. A multi-decadal oscillation in climate is a fluctuation in air pressure characteristics, wind systems, sea surface temperature, or other weather features that occur on the geographic scale of an ocean, such as throughout the North Atlantic Ocean or perhaps even the world ocean. Such fluctuations occur on the scale of a few years to decades, and need not have precisely equal fluctuations. These oscillations occur in combination with longer-term trends. As we emphasize in this chapter, our planet is experiencing a long-term warming trend that began at the end of the 19th century. It is important to realize that oscillations can cause temporary highs or lows in temperature, for example, that can partially obscure a longer-term trend. For instance, an El Niño in 1997 temporarily caused a major rise in planetary temperature. Several years after El Niño conditions subsided, one might think that the world was getting colder because we were on a downward slide of the El Niño cycle. Yes, this was true on a short timescale, the scale of the oscillation, but on a longer timescale of decades, world climate continued to warm toward the present day. The year 2016 is worldwide the warmest on record. The year 2015 is the next warmest year. Climate oscillations, or periodic fluctuations, in features such as SST or atmospheric pressure usually are captured and analyzed after long-term monotonic climate changes are detrended from the overall data. This means that a long-term increase in sea surface temperature, for example, is subtracted from the fluctuations so that residual oscillations in SST can be identified. Figure 3.1 shows a hypothetical example in which temperature is oscillating but a long-term increase also is occurring. The detrended curve shows the multi-decadal fluctuation minus the long-term increasing trend. Along with detrending, complex changes over time are often time-averaged, so small-scale fluctuations over a few years are diminished in emphasis so that larger-scale trends can be observed. Thus, we might not plot the temperature every few minutes, but instead each data point might represent the temperature averaged over the last 10 days so that we can see broader trends and distinguish them from very short-term fluctuations. Oscillations and long-term trends need not be independent. Some recent evidence suggests that ENSO oscillations have increased in the past 100 years along with overall ocean warming. Climate oscillations are irregular alternations between coupled atmospheric oceanic states that result in changes of wind systems, oceanic currents, and regional climate. World climate oscillation is partially controlled by complex shifts of coupled atmosphere-oceanic systems. Usually, these oscillations can be identified by shifts of atmospheric pressure between major regions of an ocean. For example, the El Niño Southern Oscillation comprises a major alternation of atmospheric pressure conditions between the western and central eastern tropical Pacific, which helps to change wind systems significantly. Because wind is a major driver of current systems, there is also a coupling with surface oceanic currents, which may also be coupled with vertical movements such as upwelling. The oscillations in atmospheric conditions also exert major climate fluctuations at sea and on the continents. These fluctuations are very complex and typically the alternations of state are not evenly spaced in time. There are also interactions between different oscillation systems that make changes very complex and often not well understood. Nevertheless, the fluctuations are real and observable, and the consequences of shifts in atmospheric pressure are predictable. Unfortunately, at this time there are few good explanations of how these oscillations are driven. One model of the well-known El Niño suggests that as warming occurs in the eastern Pacific, physical conditions are established that dampen the warming, setting climate into a cycle of cooling in. The Eastern Pacific. El Niño is a global-scale climate oscillation dominated by a major warming event that spreads from west to east in the tropical Pacific Ocean. As introduced in Chapter 2, northward winds along the coast of Peru are influenced by the Coriolis effect, and Ekman transport moves surface waters offshore to the left. Surface waters blown offshore are replaced by the rise of deeper nutrient-rich waters, which fuels high rates of productivity in the surface waters. El Niño, named for the Christ child, was first recognized as a Christmastime weakening of these winds. On a much larger geographic scale, the trade winds drive surface waters in the tropical Pacific toward the west. This normal condition results in deeper waters rising, which contributes to the upwelling seen in the central and eastern Pacific, bringing nutrients to the surface and helping to drive the very high productivity seen off the west coasts of tropical North and South America. El Niño develops when weakening of the trade winds persists for many months or even years. Such a persistent reversal causes a cessation of upwelling and rapid warming of surface waters in the eastern Pacific from South to North America. Not only are nutrient-rich waters no longer brought from deep water to the surface, but there are mass mortalities in fishes from the increased temperature of surface coastal waters. Warm waters often cause severe mortality of normally cold water-adapted shellfish in the intertidal region of Peru and Chile. Peter Glynn has documented extensive coral mortality in Panama during the 1982-1983 El Niño event owing to the increase of sea surface temperatures. The El Niño of 2015 caused extensive damage to coral reefs throughout the Pacific Ocean, see Chapter 17. El Niños usually involve strong coastal damage from the eastward movement of storm fronts. During the El Niño of 1982-1983, about 600 people died from intense storms, and Peru suffered fishing losses and about $2 billion in storm damage. The effects were also felt along the California coastline, and many homes slid into the sea. In a disastrous flood in Peru in 1728, the Pura River became raging rapids as a result of storms triggered by El Niño. Many people were swept away, and the survivors were mainly women who were wearing hoop skirts, which served as flotation devices by trapping air below the women's waists. In 1997-1998, El Niño was extremely damaging throughout the Pacific. During the height of this period, surface production in the equatorial Pacific was very low. But upwelling commenced again at the end of the cycle, and production of the plankton increased rapidly. Warming in the 2015 El Niño has caused coral bleaching throughout the Pacific Ocean. It used to be thought that El Niño was just a local effect along the Pacific coast of tropical America. However, a similar cycle of fluctuation of atmospheric pressure had also been recognized in the western Pacific and Indian Oceans, named the Southern Oscillation. We now know that these two pieces operate in synchrony. Hence the name El Niño Southern Oscillation, or ENSO. ENSO is driven by climatic oscillations that seem to be worldwide in extent but certainly involve an oscillation of the ocean atmosphere system across the tropical Pacific and Indian Oceans. Since the trade winds during El Niño are very weak or even reversed, the westward flowing equatorial surface current is reduced in intensity, allowing a deeper eastward flowing countercurrent to dominate. Warm water spreads eastward across the tropical Pacific and eventually reaches the eastern Pacific continents of mainly tropical North and South America. Every 2 to 7 years there is a reversal of atmospheric pressure systems across the Pacific and Indian Oceans. In the so-called normal state, the trade winds are strong and upwelling in the eastern Pacific is well developed. A persistent version of this state is known as La Niña, when there is a regional low-pressure system in the Indian Ocean and a high-pressure system over the eastern Pacific. During ENSO, rain usually increases greatly on the western coasts of the Americas, but strong droughts dominate western Pacific areas such as Australia. The effects are broader in scope than just the Pacific-Indian Ocean regions, they occur worldwide. For example, when the ENSO part of the cycle occurs in the Pacific, warmer conditions may prevail in northeast North America. It has been argued that El Niño reduces wind shear in the eastern Pacific and sets up conditions for strong hurricanes but weakens such conditions in the tropical Atlantic. The effects are even farther in the Atlantic. The North Atlantic Oscillation causes strong fluctuations in wind systems and oceanic climate. The North Atlantic Oscillation involves a periodic shift of atmospheric pressure in the North Atlantic Ocean. Normally, there is a low atmospheric pressure over Iceland at about 64 degrees north latitude and a higher atmospheric pressure over the Azores island chain at approximately 38 degrees north latitude. The difference in pressure between these two locations fluctuates. When the difference is strong, westerly winds are enhanced, and the eastern Atlantic and Europe have cool summers and mild winters. A large difference in pressure also tends to increase winter temperatures along the east coast of North America. In addition, the NAL may affect the position of the Gulf Stream, and large differences in pressure between the two locations may push the Gulf Stream more toward the north. Strong storms track eastward across the North Atlantic. During a negative NAO, the difference between pressure centers is weak, the Mediterranean region experiences more precipitation, and storms across the North Atlantic are very weak. Such large-scale climate fluctuations have been found to strongly affect local coastal conditions, leading to a revolution in our thinking of coastal fluctuations. A surprising relationship was discovered between the NAO and the western Swedish Gullmar Fjord. When the NAO is negative, winds from the north and east tend to draw dense water from the depths of the fjord, causing exchange with the North Sea. But in the past 2 decades, an increasingly strong positive NAO index has caused winds to come from the south and west, and there has been only a weak exchange of deep fjord waters with the open sea. This allows organic matter, much of which comes from human nutrient additions, to settle in the deep part of the fjord. This in turn causes low oxygen events, which are very stressful for the local deep biota, causing large-scale mortalities of fish and larger crustaceans. Winters in southern Sweden are also warmer. Increased water column stratification during positive NAO index values has also been associated with toxic algal blooms in the 1980s and 1990s. So we can again see a direct connection between an ocean-scale climate fluctuation and local environmental shifts. Before the connection was made, no one had a clue as to why these major shifts occurred in the fjord. As mentioned, the NAO winter index has shown a trend of increase over the past decades, which can readily be seen by fitting a trend over time. The oscillations behind this multi-decadal trend can be seen in the detrended plot of the NAO index in Figure 3.5b. The NAO positive index is associated with warm ocean waters in the northeastern coastal waters such as the Gulf of Maine, along with the North Sea. This warming is likely partially responsible for the failure for recovery of the cod fishery from overfishing. Cod larvae in the Gulf of Maine are at their upper thermal limits, and warming causes stratification and lower productivity, which also limits cod production. In contrast, the positive NAO index is associated with very cold temperatures in the Labrador Sea, where cod larvae are at their lower temperature limits. In this region, the very low temperatures may have contributed to the collapse of the Newfoundland cod fishery. Some recent evidence suggests that there is also a more immediate connection between availability of an important planktonic food of cod larvae and juveniles, the copepod Calanus finmarchicus. The Arctic Oscillation Index causes major alternations of climate in the North Atlantic. Its timing is strongly correlated with the North Atlantic Oscillation Index. The Arctic Oscillation Index reflects pressure shifts throughout much of the northern hemisphere, including the Arctic. In a positive phase, polar region surface pressure is relatively low and a jet stream confined to the north keeps frigid Arctic air confined to very high latitudes. In the negative phase, polar surface pressure is higher, which causes a shift of frigid air southward to middle northern latitudes. The timing of oscillations is strongly correlated with the NAO, but the AO has far broader latitudinal influence. In Chapter 19 we discuss the impact of the AO on shelf productivity patterns in the Northwest Atlantic. The Pacific Decadal Oscillation causes decadal switches in climate and may strongly affect coastal marine food webs and facilitates large movements of mobile large predators north or south along the Pacific coast of North America. Among the many other climatic fluctuations now known, the Pacific Decadal Oscillation stands out because of its strong effects on ocean climate and ocean current systems. This oscillation shifts phases every 20 to 30 years with warm waters found above 20 degrees north latitude in either the eastern or western Pacific Ocean. The oscillation is believed to be a complex combination of climate interactions, but the oscillations themselves drive strong biological fluctuations. During a cool phase in the eastern Pacific, northerly species such as Dungeness crab Metacarcinus magister and English sole Paraphrase vetulus move southward along the Pacific coast of California, and the northerly range limits of southerly species such as those living on rocky shores are truncated. Some of the strongest biological effects were first noticed in San Francisco Bay when the density of phytoplankton was found to increase. Under former conditions, filter-feeding clams and mussels were abundant in southern San Francisco Bay, and these bivalves cropped down the phytoplankton to low densities in the water column. But as regional waters cooled after 1999, Krangan shrimp, juvenile Dungeness crab, and English sole increased to record levels in San Francisco Bay and decimated the clam population, releasing the phytoplankton to increase in abundance in the water column. Thus, an ocean-wide climate oscillation strongly changed a local bay food web by facilitating the introduction of top predators. On a larger regional scale, the PDO in the eastern North Pacific is strongly correlated with the state of salmon fisheries, with an especially tight correlation in Alaskan waters. We have data on fisheries only since the 1930s, but evidence for PDO climate fluctuations go back further, even a few hundred years, based on tree ring data demonstrating climate oscillations. The Alaskan salmon fisheries catch has been positively correlated with a PDO index, which is related to increased SST. For example, in the late 1970s, Alaskan catches for all 5 species increased greatly as the PDO changed strongly to a positive phase. It is believed that this transition is due to oceanic changes in the entire food web, starting with phytoplankton, the greenhouse effect, and changing ocean climate. The greenhouse effect Earth and oceanic climate is moderated because of the greenhouse effect. The short wavelength radiation of sunlight heats the Earth and is converted to long wavelength radiation, which is absorbed by the atmosphere. Atmospheric circulation distributes this heat to space, but much is radiated back to the planetary surface, which makes the Earth's surface warmer than it otherwise would be. This is known as the greenhouse effect because of the analogous role of glass greenhouses as heat traps. Greenhouse gases absorb the infrared radiation even more and prevent it from returning to space by radiating part of the heat to the Earth's surface. Greenhouse gases, in order of their effect, include water vapor, carbon dioxide, methane, nitrous oxide, and ozone. Of other greenhouse-trapping substances, only chlorofluorocarbons, constituents of cooling fluids and some solid materials, have been nearly eliminated in production over the past few decades owing to their negative effects on the UV-protecting ozone layer. Carbon dioxide is increasing steadily, and methane is also increasing. An overall index of greenhouse gases developed by the National Oceanographic and Atmospheric Administration of the United States shows a steady increase in total greenhouse gases with no reduction in sight. See http www. Quantities of carbon dioxide have increased strongly because of the burning of fossil fuels and less so owing to deforestation. These factors are warming the Earth's surface and ocean waters. Human activities have reached a point at which they are altering the Earth's global climate. Since the Industrial Revolution in the 19th century, industrial activity has greatly accelerated the burning of fuels, particularly fossil fuels such as coal and petroleum products. Approximately 30% has been added to the storehouse of carbon dioxide in the Earth's atmosphere. Measurements from Mauna Loa, Hawaii, demonstrate an increase from 315 ppm in 1958 to 390 ppm in 2011, and it surpassed 400 ppm in 2015. It is believed that the atmospheric concentration was 280 ppm as the Industrial Revolution began in the 19th century. Most scientists believe that current trends could cause a doubling of the carbon dioxide in the atmosphere over the next 100 years. Deforestation is a secondary source of carbon release. Trees are net absorbers of carbon owing to photosynthesis, which absorbs far more carbon dioxide than is released during respiration. Many of the tropical forests in Southeast Asia, Africa, and South America were cut down to clear land for agriculture and to provide wood for sale to the developed world. Worse than that, much of the forest is burned, which releases yet more carbon dioxide into the atmosphere. Nevertheless, the impact of this release is probably less than 10% of the total carbon dioxide released owing to burning of carbon-based fuels. The problem with these releases is the heat-absorbing capacity of carbon dioxide, which radiates more and more heat to the Earth's surface. Many have predicted that continued emissions of greenhouse gases will continue global warming. There is convincing evidence from overall world temperature records that the Earth's air temperature has increased over the past 100 years by about 0.74 degrees Celsius. Because there were some decade-long periods of no increase in temperature, even though industrial burning remains steady, it is clear that global warming induced by industrial activity is not a linear process. Models evaluating human effects strongly support the hypothesis that human addition of greenhouse gases are the principal contributors to warming in the past 100 years. Warming in the coastal ocean has increased greatly, especially in the past few decades. Even a couple of degrees centigrade on average may translate into major local changes. Temperature increases may shift the latitudinal distributions of important fishery species and may even cause severe physiological stress. The magnitude and even the direction of climate change varies geographically owing to spatial differences in the effects of global warming, many of which can be predicted by climate models. Cloud cover is a major factor in climate, but it varies strongly both geographically and seasonally. Clouds consist of an aggregation of tiny water droplets or ice crystals that condense from the rising air as water vapor combines with atmospheric dust or other substances, known as aerosols. Clouds reflect short-wavelength radiation, reducing atmospheric heating, but prevent the back-radiation of long-wavelength radiation, which traps heat. On balance, clouds tend to cool the